Thank you for the invitation. I'm really glad to be here. And it has been really some very constructive days talking to the other scientists and discussing different matters. It's really a different atmosphere compared to normal scientific meetings. Well, and that is in a positive manner. Yeah, so my talk is about cellular energetics and how we will approach that in studies in ME CFS. And I'm from the Department of Biomedicine, which is very close to the Haukeland University Hospital in Bergen, where Fluge and Mella and their co-workers have been performing the clinical trials. Since 2015 or something, we have established a very close translational research environment on ME CFS. So based on the clinical trials that has been conducted, they have collected this biobank with patient samples, and together we are analyzing these samples, and we try to find biochemical changes that we would like to translate into laboratory experiments. So up until now we have been working on metabolomics, which is metabolic profiling of these samples, also some gene regulation, and we also have a study where we try to identify ME families and look for specific gene variants that can be important for the disease. And we can have also some viable cells from some patients, like PBMC, which are blood cells, and we can take fibroblasts or skin cells and culture them. And so that is in kind of in the middle here. And based on the findings in these same samples, we will use also conventional cell models and do targeted studies on mechanisms and maybe potential biomarkers. So this is how we are working. And we also got funded last year from the Norwegian Research Council on this project, defective energy metabolism in ME CFS. And I would like to say that this is also work that has been funded for some time from the Kavli Trust in Norway, and we are also very glad for the support from the ME environment, patient environments, and particularly ME-foreningen in Norway. So in this project we are asking if ME is triggered by an abnormal response that ultimately leads to a defective energy metabolism. And there are 4 different goals there. One is to continue to maintain and even maybe extending and developing the ME biobank. We want to extend the metabolic profiling of these samples. And we would like to identify risk genes based on the ME family studies by doing exome sequencing. And based on the findings, particularly in 2 and 3 here, we hope to come up with hypotheses of mechanisms and biomarkers that we can take further in laboratory research. So what we're doing, by analyzing the patient samples, then we get a snapshot of a particular point of view. And this snapshot can vary some, both in where it is pointing, but also of which resolution it has. So it is quite, when we get this data, we have to be very cautious about how we interpret them. And therefore, we do different kinds of analysis. We are looking for changes of fuels that are circulating, like sugars, carbohydrates, like proteins, which are degraded to amino acids, and also lipids. And we are also looking for specific factors that can be a result of an abnormal response, or it can cause an abnormal response. We are simply just looking for differences. And we hope that those differences can tell us something about what's going on inside the cells and if there is a particular dysfunction. And we are here, I'm just illustrating the central pathway of energy metabolism that we are coming into soon. So in 2016, we published this paper where we found a change in the pattern of the 20 amino acids. And this change in amino acid pattern in the blood samples, we linked to a dysfunction in the central axis of energy metabolism. And here it is. You can see this is glucose, which is degraded to pyruvate during this process, glycolysis. This is an anaerobic process. Pyruvate is taken up by the mitochondria and oxidized through the TCA cycle and oxidative phosphorylation or mitochondrial respiration all contribute by using oxygen and producing ATP. Also, some ATP is coming at different stages here. So based on the amino acid profile, we hypothesized that something was happening here. And it's also important when we are looking at this pathway that actually other substrates can contribute to maintain mitochondrial oxidation, and particularly amino acids and fatty acids. And they can come in at different stages here. So if that we do all the time in the lab, we try to block different pathways and see what's happening. Actually, in a normal cell, if you block one pathway, they have so many backup pathways. So you may not see very much. But we thought that this amino acid, this change in amino acid pattern could mean something, could be important. So what we think is that if there is an obstruction at this level, this will create a weakness or some kind of stress situation that may not be visible under all conditions, but there may also be some conditions where it may be visible and have critical effects. So we are investigating both the cell's abilities to adapt to such conditions and also what actually the weakness may be. So we used the seahorse technology to study this also. We also asked if there is some active component in patient blood that may mediate an intracellular metabolic change. So we cultured normal, healthy muscle cells, and we exposed them to sera from patients and healthy controls. And the take-home message from that study, which is in the same paper, was that there was a change both in oxygen consumption, which is a very important parameter for mitochondrial function, and also in lactate production. So there was something in the serum that changes muscle cell metabolism, but we don't know what this is, and it can be quite complex. So we are trying to track these mechanisms further. And now we're also using radioactivity to find out what's going on. And what we can do here is that we can take specific substrates like glucose, pyruvate, specific fatty acids, and we can label the carbons with radioactivity. Or the carbons may be radioactive, like C14, which we're using here. And by doing that, we can give each specific substrate to the cells, and then we can collect CO2, because CO2 is produced by the oxidation machinery here, not in the glycolysis, but in the mitochondria. And when we collect that, we can measure how much of the substrate which is going through the different pathways. And we have established this method now, and we are about to start the experiments towards ME on this. So this we are really much looking forward to test more. And we are also looking not only for what happens in kind of the nutrient flow through the cell, but also the factors that may regulate this, and which we know are important in many diseases. And here are just some of these which we know, for instance, are important in cancer, or also aging and neurodegeneration and diabetes. They contribute to maintain the energy metabolism and other things in the cell, and if something happens with one of these, you may have a problem. So we will try to do targeted approaches to answer if some of these factors may be involved. So these factors, they regulate a kind of a metabolic programs that can be activated by normal changes like starvation or fasting or lack of oxygen or training or metabolic stress. So you can have different types of metabolic programs. And here is, we have got this, until now we have fibroblast skin cells from one family which is ME affected. And in such families we can of course also look for possible mutations that may cause the disease, and we are also doing that. What we did here was to measure oxygen consumption of these fibroblasts and under different nutrient conditions. And we think we are seeing some differences in the ability to adapt to different conditions. So we are thinking that something may sit in the sensory system that mediates an adaptation to a nutritional change. So this we will look further into. But it's also important to note that this is not the optimal cells to look for metabolic changes from patients, but it is the cells we get most easily from patients, right? And we can have, at least that we can have in the laboratory for some time. So we have until now mostly worked with PBMCs from the blood, which is very easy to get, but you cannot maintain those either in the laboratory. You need to have fresh samples to really do the study. So it's quite big experiments to just do that also. But we do use some muscle cells which are commercially available, and then we try to make or mimic or target specific mechanisms in those. So we have to have some different ways to do this. Now I will take you through some cartoons about how we are thinking, our concepts regarding this. And just to take you back to some basic lessons of cell biology. So the energy output needs to be balanced by the fuel input, right? And all this is mediated, or maintaining the cellular energy status is mediated by the cell metabolism. So the cellular energy level is kept quite constant under variable conditions, and this is the concept of homeostasis. And metabolism is also balanced to the fuel supply and workload. So all these contribute, and the cells cannot do more work than the energy level permits. So the workload is also determined by the energy capacity, and then particularly of the mitochondria. And what kind of threats may there be to this? Well, you can have reduced fueling or reduced level of oxygen. You may have an increased energy costs or energy leak or a metabolic obstruction of some kind or disturbed control. There are several things, and these are overlapping also. And they may all contribute. And of course, the cellular energy demand will also depend on different conditions. This was a little bit busy to look at, so I make this static one. But so the energy fueling needs to be balanced by the workload. So under work, you need more fueling. But also there may be shifts between the different fueling systems or pathways. It's important to have this in order to maintain the capacity of the system. Under recovery, there may be some increased fuel requirements, some increased energy requirements, but there might also be some increased or some different waste, getting rid of waste processes. So, and in order to get back to rest, you need to do this. And this is, yeah. So I will just use this as to try to illustrate how we are thinking maybe about a very simple ME model. We found a metabolic obstruction or indications of that, we think, and how can we address that in our research? Well, if we have this obstruction, the cell will need to use an alternative fuel source, right? It may not be the first choice, maybe it's the second choice. And this may lead to some kind of metabolic erosion or weakening of the system. So the cells here try to have some kind of damage control, and maybe the system is not really resting, and this should be on the resting phase compared to the normal situation. So what if you add some more energy requirements to the system? Now the cells are working, and you really have increased requirement for fuels as well, but since the cell may not be able to fully use the system, there may be a lower threshold for overcharging this, and if you overcharge it, you will have a lowering of the cellular energy level, and this is not sustainable. They cannot be sustainable. So then the cells has to do something to reduce the energy requirement. And here there would be really a struggle for energy. And when the cell then would try to recover again, it may not even here use the first choice pathways. It may have to use alternatives. So they may be less efficient, there may be more to restore under this condition because it's more stressful for these cells compared to the normal, and there may also be a waste problem. So there's a higher cost here, and maybe even a cellular post-exertional malaise. We have not been able to find a marker for that, but we will look. So how we are working is that we try to mimic and target these different conditions and mechanisms in cell cultures. We can use conventional cell models, and I guess you all have heard of the HeLa model, which is the most commonly used. There are many others. And we can also link it to other known mechanisms. So we will investigate these mechanisms, and we will target them using different kinds of manipulations, both pharmacological, nutritional, and also genetic. And we can also get these samples from patients that we can use to have a translational approach in the laboratory. And of course, yeah, there are many links to other research fields that we can benefit from. And how we can try to mimic or create stressful situations for the cells in the culture. We can put them under some kind of nutrient deprivation, or we can replace nutrients. We can make restrictions for the total energy supply. We can control oxygen level. But we can also use targeted strategies to really modulate specific metabolic programs. We can do it pharmacologically. We can also do it by doing some genetic modifications. And typical programs would be to create some kind of pseudo-starvation or pseudo-hypoxia, and force the cell to do some kind of metabolic adaptation or even chemical training. And here we are now. So the nutritional manipulations would be at this level, whereas changing these programs would be at the sensory system. And we can also, of course, add specific inhibitors to specific enzymes and metabolic stressors, and also mitochondrial uncoupler, which is a creates an energy leak in the system. So our conclusions are that there is a struggle for energy in these cells, and they have a suboptimal way of suboptimal metabolism. And this may create a weakness that may not be visible under every condition. We think that these mechanisms are reversible, and we are now working to identify these cellular weaknesses. We want to identify mechanisms, and also to contribute to making good models for ME/CFS in the laboratory, and of course to search for biomarkers. I will end this by thanking the research environment in Bergen for creating such a good environment for do ME/CFS research there. Thank you very much.
Thank you, Karl. Some questions, please.
Given that the mitochondria really seem to be the focus of attention here, and that they seem to be dysfunctional in some way or other, have you thought of comparing mitochondrial genome sequences in affected patients with the same sequences in siblings who share the same mother, given that the maternal line is the way the mitochondria are inherited?
That would be an interesting approach, and still, I think some aspects may be addressed, but I think if you're looking for mechanisms, it is a quite complicated way to go, because the mitochondria only encodes for 13 proteins which belong to the respiratory system, and if you have say a very severe genetic defect there, you will see it in different means. So and also you have this. There are multiple types of copies. There may be there is something called heteroplasmy, which means that you have a mix of different genotypes of the mitochondrial genome. So we have not gone there yet, but it might be in the future, right? I don't know if somebody else have.
Yes, exactly. Is there anybody else in the audience knows of any work on that front going on? Mitochondrial DNA?
Yeah, Hansen, no?
It's Maureen about,
yeah.
She might.
We've published a paper showing that after sequencing 200 patients and 200 controls mitochondrial DNA, that we did not find any differences.
Right.
Okay.
That were correlated with having the disease or not. Now, we did find some differences that were correlated with severity of certain symptoms, but not with having the disease or not. And we didn't find anyone with a genuine mitochondrial disease among the patient population, although there was one control who has a mitochondrial disease but doesn't know it, according to the genetics.
Thank you.
Thank you very much. Any other question? Final question?
Paul Watton, do you have any thoughts on mitochondrial supplementation, like NT factor, etc.? Just trying to load the system to give it all the nutrients that it could possibly want. Have you thought along those lines? Is that in your plans to look at that kind of thing?
So what we have to, our support for doing those kind of things is actually what we see in the metabolite profiles if we see some kind of deficiencies or reduced levels. I think it's difficult to come with any recommendations there, because I know there are various individual experiences by having, and many have tested and have had benefit, but from our point of view we have no support for advising anything about mitochondrial targeted treatments, so to speak. Okay.
Okay, well thank you very much, Karl. Please come and join us and we'll try and get some questions in at the end.